about increase in substrate binding or decrease in substrate binding. So in essence, it can increase the reaction or it will suppress the reaction or inhibit the reaction, whichever, okay? And then finally, ubiquitination. Ubiquitin is very common in the cell and it plays a role in the degradation of the amino acid, okay? So there are two types of how to degrade the amino acid or the protein. And then after that, there will be this degradation further to release all the amino acids. So this picture is telling you that the amino acid, the protein that has been degraded to its component amino acid can become a substrate for translation to form another protein. And that is the life cycle of the cell protein. By the way, we need the 20 amino acid, and if this is the selenoprotein, then it will happen during translation to give us the selenoprotein. Okay, so the 20 amino acid is required for translation. So primary structure is lipid, and it is also referred to as covalent structure. Anyone who can tell me why it is also referred to as the covalent structure? Do you understand by covalent bond and non-covalent bond? Sequence, the definition is refers to the linear sequence of the amino acid. So one amino acid from the N terminus, then followed by the next, the next, the next, the next, the next, until it reached the C terminus. So the question now is, what connect the amino acid together? And the answer is, anyone? So it's what? You should know. Okay, peptide bond. So peptide bond is a covalent bond. Okay, that is why it is also called the covalent structure, because there is no other bond that is happening there in the primary structure except for covalent, the peptide bond that connect the sequence of amino acid. Okay. So this is just to show you that condensation will follow. There is also a concomitant release of water in condensation reaction. Okay, let's take a closer look at the other bonds, okay, that are present in the polypeptide backbone or in the polypeptide chain. So we mentioned already that the peptide bond is a partial double bond character, an amide bond, wherein there is limitation of movement or rotation. But there are these other two bonds known as the psi and phi, or the phi and psi. So the black one is between alpha carbon and the carbonyl carbon. The red one is between the N carbon and the alpha N and the alpha carbon. Okay, so these two can undergo rotation. The phi angle and the psi angle, so they can twist. So much so that this famous biochemist known as Ramachandran was able to come up with what is known as your conformational map. So he will be able to plot the psi and phi angle. By doing so, there will be a repeating values that is common to either the psi or the phi. So depending on the value, he will be able to predict whether this is an alpha helix or whether it is a beta sheet. I don't expect you to memorize the number, okay? So basing on what is the range or the common value that is observed, he can plot it out and he can predict that the predominant secondary structure is either alpha helix or beta sheet. So for you, okay, we go back to primary, it is linear straight. Then something will happen within that linear structure. So there will be a local ordered structure brought about by specific backbone torsion angles and hydrogen bonding. So what brings about these torsion angles are referring to the psi and phi angles, okay? And that eventually what stabilizes the secondary structure are the hydrogen bonding within the peptide backbone. Can you picture that? The helical. That's why it can twist and it can connect. So the stabilization here, in addition to the peptide bond that you see in the primary, it can now conform to another conformation by the twist of the psi and phi angle, and then it will be stabilized by hydrogen bonding found on the peptide backbone. So meaning to say the carbon that is part of the peptide bond is the one that will stabilize it, 4 residue apart. And we mentioned that the most common is either alpha helix or beta sheet or beta plated sheet, okay? So what you are seeing, the one on is very obvious, the green one, which is helical, and the other one, you see the arrow going down and then going up. This is the anti-parallel beta plated sheet. If the direction is the same, if both are going up, then these are parallel beta plated sheet. Okay. So all of this is in the book. Okay. So first, it is helical, is right-handed or clockwise direction. There are 3.6 amino acid per helix turn. Then the R group is found extended outward. Okay. Then take note of the carbonyl group. Okay, the C double bond also that will indicate that this is the carbonyl group of the carbon. Will now be also parallel to the axis of the helix, and it will hydrogen bond to the NH group, okay, of the peptide bond for amino acids below. If you can see the, excuse me, the picture wherein it will show the hydrogen bonding, it is the dot, the red, and then the dot that is connected to another atom, which is for residue away from the one above. Okay. So these are the hydrogen bond. Now I put there a group of, okay, anybody who can tell me what is aspartate and glutamate and what is isoleucine, leucine, and methionine. These are your
non-polar.
Non-polar.
No.
They are non-polar. How about ALA? Which is non-polar? The last 3 is non-polar, right? ALA is also non-polar. Aspartate and glutamate are polar. Okay. Then you have your glycine and also the proline, which we explained. This is where it will be found because it is the smallest, and the pyrrolidine ring will actually have a difficult position. It will provide the turn possible that you see on the structure of the molecule. The important thing, what is amphipathic? So we said it is, the R group is on one side, okay? So on one side, it can be all hydrophobic. On the other side, it can be all hydrophilic. So the amphipathic, meaning to say polar and non-polar, is present. Just like your phospholipid is also amphipathic. It has a polar head and a non-polar tail. So this can actually form interfaces between polar and non-polar regions, so much so that it can play an important role in your formation of clusters of amphipathic helices, can create channel or pore in the membrane. And that is how your transport membrane protein are formed. Okay, so it will allow polar to come into the cell. The next is the beta platelet sheet. Okay, this time, where is the positioning now of your R group? It is above or below. So it forms 2 or more different region or stretch of at least 5 to 10 amino acids. Okay. So beta platelet sheet should at least be 5 to 10 amino acids. So it can be more than 10. So that's why, as shown in the illustration, it will form a platelet sheet. And it is anti-parallel or parallel. Then we also have what we call the, so there are many types of secondary that can be conformation, but the most important or common one is the alpha helix and the beta platelet sheet. We also what we call the beta turns or bend and the loops or the random loop, as noticed in the illustration. The red one is the beta bend, and then the black one are your loops. So the beta loop, approximately 1/3 of all the residue in globular protein are contained in the liver. When it turn out to form the tertiary structure, so they will be turned. They will become more compact. The chain will fold sharply back upon itself to reverse the direction of the polypeptide chain. So 4 residues are present, and again, the stabilization is by hydrogen bonding. So primary is peptide bond, secondary primarily is through hydrogen bonding, and then we also have the proline and glycine playing its role. Okay. This is the super secondary structure, or referred to as the protein motif. The motif that is found in the protein. So some protein contain an ordered organization of the secondary structure that form a distinct functional structural protein. So these are super secondary structure. These are functional structural motif. Example given here are the 3 kinds of transcriptional regulators. So these are protein that has the capacity to bind to your DNA or RNA. Okay. The one above in blue is your zinc finger. The one below it is your leucine zipper. Okay, so it looks like a zipper. And then the one next to it is your helix loop helix. So let's say in the future, if you are into research and lo and behold, eureka, you discovered a new protein. And and it has this more or less conformation, it is safe to say that this kind of protein has DNA binding capacity and can act on the DNA. So these are DNA binding protein motif. Tertiary is the global folding, okay, of the single polypeptide chain, or in other words, you think of a three-dimensional form or three-dimensional structure. Then you will ask yourself, what is the question that you will ask? We said primary, stabilized by blank. Secondary, stabilized by blank. So this time we now have a tertiary structure best exemplified by a globular protein. So you should also ask the question, what is responsible for the bond that will allow it to form a tertiary structure? Anyone?
Non-covalent interactions daw.
Yes, non-covalent interaction involving blank of the amino acid.
R groups.
Very good. So this is the one that we did not talk about yet. So the R group can come together, interact with each other, okay, non-covalently to assume the tertiary structure. So what are the non-covalent bonds? We mentioned hydrogen already, right? So this can happen. Van der Waals is always there in the molecule. So what else? What did you learn about structure while we are in the discussion about amino acid?
Disulfide bonds.
Disulfide is a covalent. It is correct that disulfide bonds will be present in the tertiary structure, but it involved, again, it is correct, it involved the R group, but it is not a non-covalent, it is a covalent bond. Okay.
Electrostatic
Very
interactions.
good. Electrostatic is between,
Positively charged and negatively charged
yes,
groups.
the positive and negative R group. What is the other name? There are two other names of electrostatic.
Salt bridges.
Very good. Another one. They have charge, so they are ionic. So ionic bond. So electrostatic, ionic bond, or salt bridges, they mean the same thing. An interaction between positive and negative ions found on the R group. Okay. And don't forget about the hydrophobic effect. Where are they found? In the globular protein, in the interior of the globular protein. Okay. This is also the answer asked by Dr. Pipito about how the, how the cell, can come together as to assume each membrane is found hovering outside the cell. This is because of the hydrophobic effect of the phospholipid, wherein the two tails, if you look at the structure of the cell membrane, the two tails are connected to each other. So this is the hydrophobic effect. So the driving force that will make it globular is the presence of the hydrophobic amino acid coming together through an interaction. Okay. Then we have, what is the meaning of a domain? A domain is a section of the protein structure sufficient to perform a particular chemical or physical task. Okay. So we call it a domain already when it assumes the tertiary structure, if it has a function or a task. And then we can call it a motif. If structurally you've seen something, then you can predict that it can perform a particular function. Okay. So binding of a substrate or a ligand. So meaning to say, in the receptor, there is the binding domain to the ligand that will allow the ligand to interact with it so it can bind. Or it can anchor a protein to a membrane, or it can interact with a regulatory molecule that will modulate the function. So quaternary. More than one polypeptide chain is called a quaternary structure. Okay. So you have two or more polypeptide chains. Each of the polypeptide is known as the oligomeric protein, or you have your monomers or the protomers. Not to be confused, we will just call it more than two polypeptide chains. So not all protein exhibit as quaternary structure. Meaning to say, not all protein can have more than one polypeptide chain. So with this statement, what is it telling us? Protein become functional in its quaternary, at least it has to be in its tertiary structure, and for other, it has to be in the quaternary structure. Is that clear? Okay. So all of this is brought about by your after the primary, the rest of the structural form or conformation are brought about by your non-covalent interaction. Okay. Okay. So this is your Ig molecule. How many polypeptide chains do you have? You have the blue one, two of them, and the red one, two of them. And what do you see? Within the blue polypeptide chain, because of the tertiary conformation, you have the disulfide intra chain. You also have the disulfide that is inter between the two chains. Get it? Okay. I have presented this because I said before, in some biochemistry book, they also refer to a quintenary, but it does not mean that there are 5 polypeptide chains. A quintenary structure which refer to your complex protein, and this is best exemplified by your pyruvate dehydrogenase. So, okay, let's take a look at the red molecule written on the left, which is a pyruvate. And then what you have in the first one is your pyruvate dehydrogenase, that is one enzyme. Another enzyme is your dihydro, it's very small, dihydrolipoamide dehydrogenase, and then lastly is the hydroxylipoamide dehydrogenase. Okay, so there are actually 3. Something is wrong here. There are 3 enzymes and 5 coenzymes, but their action is to convert pyruvate to acetyl-CoA. Okay, so far I have only know, from what I know, there are only 3, 4, 5 quintenary structure. And you will know as you go along in the lesson. Okay, so this is just to give you an example of your quintenary structure of your protein. 3 enzymes or 3 protein acting together with their coenzymes to catalyze one reaction, and that is the oxidative decarboxylation of pyruvate to alpha your acetyl coenzyme. Okay, so this is just to show you the hydrophobic interaction, the hydrogen bonding, the ionic bond, and then the disulfide bond. Another, okay, very beautiful images. Ionic bond, hydrogen bonding, hydrophobic effect, and then the disulfide. So in short, this is the, if you want to take picture, take this picture. So the sequence of amino acid in a protein is determined by the sequence of the nucleotide in the gene encoding it. Okay. So it depends on the informational molecule, which is the DNA. And then the shape is determined by the primary structure. Depending on what amino acid is in sequence, it will allow the R group that is found in the sequence to interact in such manner to be able to give you the tertiary structure. And if you have more than one, then it will become your quaternary protein. Okay, then the function of a protein, of course, has something to do with its shape, and the function is absolutely dependent on its three-dimensional structure, or the tertiary as well as the quaternary, if there are more than one polypeptide chain found in that protein. Okay. So we will now go to protein folding. So what is protein folding? This is a process. It is ordered and it has many steps. So if you look at the figure, so first you have the sequence, no definite form. And finally, you see that you have the secondary structure formed. So local secondary structures will be formed. Then they will aggregate. Okay, so there will be domain assembly, and then it will become your molten globule. So the tertiary structure is the molten globule. Okay. And if this is a single polypeptide chain protein, then it will end at the conformational adjustment of that polypeptide chain. Okay. But if this is a quaternary structure protein, then there should be the continuation of the association of two or more monomers, and then final adjustment of the dimeric protein. So this becomes dimeric because there are only two monomers or two polypeptide chains. Do you follow the step? Now, protein folding will require certain protein also to help the folding process. Then they are generically referred to as your chaperones. Chaperones protein is involved in the proper folding of the protein. Okay, so the HSP stands for heat shock protein. If you look at the figure on the side, you have your heat shock protein 70, and there is the heat shock protein 40 that attached to it. So meaning to say, for heat shock protein 70 to be well functional, you also need the assistant. This is sort of a co-factor for the 70. Okay. So what is the function of the HSP 70? It binds short sequences of hydrophobic amino acid in the newly formed polypeptide chain. It has to do that so that it will shield them from the solvent. One. And second, it will prevent them to come together right away. So prevent aggregation and to shield them from the solvent, thus providing the opportunity for the formation of the appropriate secondary structure. So then, Qualis, to form the tertiary structure. Okay? So that is the big function of your chaperones. The other chaperones is what we call your chaperonin or your 60S. 60 HSP 60. It has an 80 phase activity, but when I Google the HSP 70, it also has the 80 phase activity. I think energy is needed so that it will prevent the aggregation from happening by shielding it. So together with the 70, so it has a donut shape. Okay, what will happen is it will allow it to continue to fold. The hydrophobic region will be buried in the interior, thereby eliminating your aggregation. Okay? Because if there are not, if the chaperones are not present, the tendency is they will come together and it will not allow the proper conformational changes that should happen to that protein. Okay, but the ultimate, as we said, is the how the sequence of amino acid, which is the primary structure that will dictate as to the correct shape that it should assume. Okay. Now we have these other two enzymes, the protein disulfide isomerase and the proline cis trans isomerase. Okay. I told you that the disulfide will come together by interaction of the cysteine residues. So why do we have an isomerase? So meaning to say, maybe the disulfide bond that was formed first is not the right one, so it will attach, okay, then it will seek another cysteine until finally it arrive at the correct cysteine that should come together. That is the meaning why you have the isomerase. And the cis trans is about the peptide bond that we see. Most of the peptide bond is in the trans configuration, but for proline, because of the ring structure, it is easier for it to form a peptide bond in the cis configuration. So all of this is needed to eventually assume the tertiary, correct tertiary structure. Okay, and then if there are more than one of them, then it can become your quaternary structure, polypeptide. What is a prion disease? Spongiform encephalopathies or the TSE. So it has a different, there are a lot of, for the human, it is known as your Creutzfeldt-Jakob disease. So this must be the name of the doctor who discovered the disease. Okay. So we can get this from cattle when we eat our beef that is not cooked very well, and it has the prion, abnormal prion protein. And then this is actually, I actually was able to read a whole book on this. It's very interesting. Okay. The guru is people in New Guinea, Papua New Guinea before who actually practice cannibalism. And this, if you eat without cooking properly, again, the flesh, the one who eat it will finally also suffer from spongiform encephalopathy. Okay. So much so that for a long time, they wanted to identify what is the viral agent because it seems to be infectious. If you eat somebody that has this abnormal protein, you yourself eventually will suffer from the same disease. So they were thinking this must be, but it is smaller than the virus. Okay. And then finally, Dr. Stanley Bresinair, the one who was able to determine the pathogenesis of this disorder, and he received a Nobel Prize in 1997. So there are actually two types of prion protein. The C stands for cellular prion protein. So this is the normal prion protein, which is found in the membrane. So transmembrane glycoprotein found in certain cells like your neural cells, okay, or your hematopoietic stem cells. So it will affect your brain cells because that is where there is a lot of neurons in the brain. Well, the abnormal one is referred to as the SC, which stands for Scrappis, which is again an encephalopathy that is found in the lower animal, somewhat related to the cattle, which are the goat. Okay, so PC is cellular, then P scrappy, SC is abnormal. Okay, and what is the culprit? What happened? It is just a change in the secondary structure of the protein. Okay, so normally the cellular prion cell is dominated with an alpha helices as its secondary structure. It is easily soluble because it is globular and can be digested. When it is transformed to the abnormal one, the secondary somehow become the beta sheets. Okay, so this is actually smaller than the virus. This is actually about the conformational structure of the secondary structure of the protein. Okay. So what happens is if there is a molecule of the abnormal protein, it will become the seed that will provide as a template for the normal to be converted to become more abnormal. So they can form aggregates and they have the neurofibrillar tangle. This is the pathogenic abnormality seen as well as the amyloid that finally can be detected also in the individual. Okay. And this also the amyloid deposit. These are actually protein aggregates that can be seen in AD, Alzheimer's disease, as well as Huntington disease. So these two are also neurological. The possible culprit or the theory here is about the failure of the folding process. Okay. So meaning to say folding was not completely happening, so much so that it can change its secondary structure itself. Now for the AD, I think the book mentioned as to a mediator, which is a lipoprotein. So it is for you, I think I have also mentioned that AD. So what is implicated here is the apolipoprotein E, okay, which is an apoprotein in your LDL, low density lipoprotein. Okay, the main cause is elusive, but the senile plaque and neurofibrillar bundle will aggregate to form your alpha amyloid. This is actually endogenous. We need to say alpha amyloid is normally present in the brain tissue, but if that protein undergo refolding and misfolding, then it will lead to your from alpha helical to become beta plated sheet, and it can further aggregate. So it will lead to your AD, okay, or Alzheimer's disease. Second to the last is about denaturation of protein. So the covalent band or the peptide bands are what? Stable under ordinary conditions. So they are difficult to break, but it can happen, okay, if the temperature is very high and you add also a strong acid and a base. So with heating with strong acid and a base, eventually it will also break the peptide band. But that cannot happen in the human system. Okay? And cleavage of the disulfide bind would require a reducing or oxidizing agent. So denaturation of body protein can happen under certain conditions. Change in pH, change in salt concentration, and change in temperature. And that is why, as doctor, you have to know what is the normal pH value. So there is a danger of the function of the protein in our body if there is a shift of the 37 higher or lower. Also a shift of our acid-base balance towards acidity or towards alkalinity. And that is the reason why these are important. Because application here is a lot of things is going haywire inside the body if denaturation of the protein enzymes will happen. You now understand why it is important, okay? Knowledge why that is important, because what will happen, it will unfold the protein because it can affect the non-covalent band that allow the protein to assume its native conformation in the tertiary state or in the quaternary state. And the effect is it will lose its function and it is susceptible to enzyme action or the hydrolytic action. So it cannot function anymore what it is supposed to function inside the cell. In the purification, just like you to understand what is the principle here. You are not going. Sorry, it's very noisy. So we will allow the garbage truck to. So often, ultracentrifugation also will depend on the solvent used and the speed of the centrifuge that will allow it to separate. The size exclusion is about the stock radius of the molecules. So depending on the size, you can separate one from the other because your field there is the polymer inside the column of the chromatographic tube has the bead has hole, so it can navigate itself. So the smaller size will be the one that will come out last because it has to pass through the hole before it can get into the bottom of the column. Ion exchange is about positive and negative charge. Affinity is about you try to remember that the receptor has a specific affinity to a certain ligand, so they can bind together. So if you want to elucidate the one that you want to remove or find out, then that is the one you can manipulate the solvent where they are, then that will be the one that will be emitted last. High performance liquid chromatography, the HP can stand for high performance because you will require high pressure to be able to elute it out. Okay. Now SCS phase, sodium dodecyl sulfate. Okay. E, the last E is what? Electrophoresis. So do not be misled by the last E, which stands for electrophoresis, because this is not based on charge. This is based on size. Because the STS can convert all the protein that you want to separate into negative. So it is not about the charge, but all the negative protein will move toward the positive end of the electrophoresis. And of course, the smaller one can move faster than the bigger one. Okay, then electrophoresis, and then lastly is isoelectric focusing. As the name implies, it will be able to separate protein from each other or purify the protein based on the PI value in the field. Okay, so we will go very quickly. Okay. So it says here, depending on the mass. Never mind about the what solution you need to put it there. Okay. So it is depending on the mass. And then size or the gel filtration or gel permeation chromatography. It means the same thing. It has a lot of other names. Size or molecular exclusion chromatography, gel filtration, gel permeation chromatography. It is based on the Stokes radius of the molecule. As you will notice, the last one that will come out are the smallest one, because it has to navigate into the hole that is present in the resin, porous beads. And this is ion. You can manipulate the solution used. Okay. Then you know that whether you will use a cation exchanger that will attract negative or an ion exchanger that will attract, cation exchanger that, yeah, right, that will attract negative, and an ion that will attract your cation will attract the positive because sulfate is the base. But the anion, it is base, so it will attract the negative. Okay, so positive the negative. So again, by manipulating the pH of the solution, you know that if at an acidic environment, it will be what? Positively charged. But if you want to elute one, you can change the pH by knowing what is the pK of that particular protein. Okay. Affinity. As you will notice in the illustration, you have a ligand that will attract something. Okay. Let's just say that is a receptor. And you want to attract it want actually to get your shape, the red one. So that will be the one that will be eluted last. As you see in the last one, it is the red one that will appear. So based on affinity. Then high performance is either size or charge. The difference only is the column chromatography. It is smaller but more tightly packed with the resin. Because it is tightly packed, you need to have high pressure to be able to separate them based on the charge or on the size. And that is electrophoresis based on charge, and this is SDS, is based on size. So the smaller, because as I said, the protein is coupled to your SDS and they will be negatively charged. So all will move towards the anode, and it is the smaller one that can travel faster. Then this is your isoelectric focusing. As you will notice, the protein will become stationary in its PI. Okay. The last one is, this is in the book. You have the two-dimensional electrophoresis. Okay. This is a combination of the IEF and SDS page. First is you do the IEF, okay, and then second you partition it further using the SDS electrophoresis. And that is how it will look like. Okay, any question? Dr. Pipito, are you still there?
Yes, Doc, I'm here, Doc.
I am done. I don't
Any
think
questions from the students?
Excuse me, Doc.
John Mary, yes.
Regarding the Alzheimer's disease, Doc,
Oh.
at your PowerPoint, Doc, it's alpha, then at Harper's, it's beta.
No, the alpha become beta.
Okay, okay. Thank you, Doc.
Okay, normally there is no disease, so there is no AD. And then when there is increase, because the mediator that has been alluded to is your apoprotein E, which is part of your LDL actually. Okay, so that is the protein in the LDL. But the pathogenesis of the disease, when I was having my some study for my masteral in biochem, the professor told us, I am the only student, okay, very lonely. The professor said at that time, somehow the receptor, a lot of receptors found in the cell has been identified. So he is saying that somehow the disease, the abnormality is about the receptor. Whether there is binding or whether it can bind, that's the first one. Whether it can do the signal transduction process or having an effect inside the cell is another one that is disease. Or whether it can do the duty, do the number one and number two, but number three is not happening, then that can be part of the disorder. That is what he was saying. It is all about something happening on the receptor of the cell. But now, the general pathogenesis of diseases has been alluded to the inflammatory reaction, about the cytokines, whether it is pro-inflammatory or anti-inflammatory, all about inflammatory reaction happening inside the body. Even AD, I have read article that this is also caused by inflammation happening inside. Okay, so the verification, as I said, don't bother to know how it is done, know the principle. What is the principle behind the scientists being able to purify the protein from a mixture or to separate them from each other? Okay.
Any question? None?
Hello, Doc.
Yeah.
It's Josh or we. I had a quick question about the Google Calendar. It said that tomorrow was going to be the PBL instead. Is that
Yes,
true?
Josh, yes. So any questions before I proceed to announcements and reviewing your long exam this morning? other than Josh's question? Any concerns for Dr. Meneses? None? So yes, we will be exchanging the schedule for team-based learning. So your team-based learning will be on Thursday, and then your small group discussion will be tomorrow. So the Google Calendar schedule is correct. Okay.
Okay. Hey, Doc.
And
So,
then, yes, Doc Meneses?
yeah, I cannot see the stop share here. You are, okay, I have that.
Okay, sige. So next is, for those who did not get your scores, I cannot give it to you. I can only give it to you once I post or give you like a summative or evaluation score. I'll try because it's not final yet. Okay. So scores will be given to you after the exam for those who took the exam this morning. But you will have definitely get your grades before the bi-monthlies. Okay. Sige. So we will do rundown of the exam, and then I'll say the answer. And for any clarification to a particular item, Dr. Meneses is here for her questions. The other faculty are not here, but I can relate to them your specific concern to a specific question. Is Ado here?
Yes, Doc.
Rhea, you did not upload pa, no? Or you've uploaded your exam already?
I uploaded na po, Doc.
Okay, first question, which of the following peptides is referred to as the opioid peptides? What's your answer? Anyone?
Enkephalines, Doc.
So enkephalines and endorphins. Okay, next question. The presence of proteinaceous substances was found in a meteorite. If the scientists would like to determine the components of the amino acid residues present, the best solution is to subject the substance to
Anhydrine, Doc.
Ron. What's your answer?
Anhydrine.
How about you,
Another answer.
Harvey?
I answered stereochemistry.
Okay, so the correct answer to this is to determine the chemical nature of the R groups.
Okay, clarification. Can I be heard?
Yes, Doc.
Okay. The ninhydrine and the fluoriscamine is to determine the presence of protein, not amino acid. Okay? Basically, that is how it is. You cannot differentiate the amino acid from one another. You can differentiate the amino acid because I said they want to find out the component amino acid. So the component amino acid will differentiate one from the other on the R group. And then the stereochemistry is about being able to polarize the light either to the right or to the left, and also whether the isomerism is L or D. And that is about stereochemistry. Okay, so this is a question that will let you think about what is the important substituent that is present in the amino acid for you to name it either as charged or uncharged, or specifically to actually know the amino acid, whether it is glutamate, whether it is isoleucine or whatnot. Okay, so the question may sound difficult, but if you read it carefully, you will actually know the answer. Okay, next.
Next, which of the following is least likely a function of glutamyl cystinyl glycine? Answer. Anyone? Learn. This is one of the difficult. Less than 30% got this one. Pero 9 correct. The
In the
answer
dark
is
talk, reductant.
a vasoactive tripeptide.
Okay, when you say least, meaning to say it cannot function as such. Because I have to ask for the least because the glutathione has a lot of functions, right? And one of that is it is just a tripeptide, so it cannot function actually as a vasoactive peptide. It's too small. That's why the function is more of molecular.
Okay, question 4. In amino acid electrophoresis, all but one of the following amino acids remain stationary. Which is the odd one? Any answer? None? Okay, answer to, sige. Try, anyone?
So what is the principal man of electrophoresis? Going to the positive end if it is negative, going to the negative end if it is positive, and it cannot move if it is hydrophobic. So you look for the set that is hydrophobic.
So the answer to this is lysine. Okay. Next question. What is the charge of a decapeptide at its isoelectric point? Answer. Any answer?
Neutral
No.
charge.
No charge. No charge.
The answer or
No net charge.
neutral or no net charge. So that's the answer, ha? Okay. So next question. Which of the 3 letters designation referred to a derived amino acid? What's your answer? And it's one of the difficult questions.
GL,
Yes.
GLA is what?
Glutamic acid.
It is carboxyl glutamic acid.
So that's
there
the answer.
GLA, because you know SEC is not derived. It is referred to as the 21st amino acid. I emphasize on this by I said SEC will happen during translation. So this is not post-translational. So it is not derived. Because okay. Because if you know SEC, then it becomes difficult because I put both and neither. So your choice is actually 50%.
Okay, next question. So basing on the pK values of the amino acids, can you predict the charge of amino acids when they are put in an environment wherein its pH is gradually increased to become higher than the pK values of the amino acids? So your answer, anyone? So answer to this is amino acids will be negatively charged. Okay. Next question. Which of the following best describes the amino acids? Answer to this one is the charge of any proteins at physiologic pH will depend on the R groups of the amino acyl residues. Next item. Which set of amino acyl residues in the active site of protein enzymes will act as a general acid or base catalysis? Sige. Any of the combination that you can remember is your answer to this one.
Lysine, arginine, histidine, alanine daw.
Okay, so that one. So lysine, arginine, histidine, alanine. That's the correct answer. Next item, basing on the principle of paper chromatography when 4 different amino acids are applied. Which of the amino acid spot travels the least distance from the starting point? What's your answer? Answer, GLU.
Why glutamate? Because it has a negative charge because it is polar. The one that will travel farthest away should be hydrophobic.
Okay, another question that seems difficult to you. Which of the following best describes the peptide bonds found in peptides or proteins? So what's your answer to this? Anyone? Description of peptide bonds in peptides or proteins. The answer is the bond has partial double bond character and is rigid.
Excuse me, Doc. Can I clarify that regarding that question, Doc?
Okay, Queen.
Is it not semi-rigid, Doc?
And what is your answer? Is there a best answer aside from the answer?
I think
You should,
What's
you
your
should
answer?
not be you should not be looking for the answer that is not there.
So what's your answer, Queen?
So what's your answer?
I think I just answered the one with elimination of water, Doc.
No, because condensation will happen every time there is linkage in all linkage of the macromolecule through condensation or putting them together will always have elimination of water. So that is not the best answer. Because we're talking about the bond. The emphasis is on the bond. You get the idea?
Okay, Doc. Thank you, Doc.
Okay.
Okay, next question. Which amino acids in protein has the ability to strongly absorb UV light at 280 nm?
This is a giveaway.
Tryptophan.
Tryptophan daw.
TRP or tryptophan. So that's the answer. Which of the following reactions does not occur in amino acids? Can you remember your answer? So does not occur in amino acid. Answer, disulfide bond formation between two MET. I'll proceed. What is the reason why hemoglobin can act as a buffer?
Because of the histidyl content daw.
So what is in histidyl content?
PK6 daw.
Yes, it's the pK value that matters.
Okay, so that's the answer, the pK value of imidazole group of histidine.
That is the last one.
Yes, that's the last of your questions, Doc.
Okay.
You can go ahead.
And as you notice, all of the questions pertain to the characteristic of the amino acid. Is it not? All. And there are questions about the peptide, of course, maybe I think one question or two, but everything is about the amino acid, okay? And I specifically ask you to understand or know your 3-letter designation of amino acid. If that is the one that you are confused about, then that is, I'm just sorry if you did not try to understand. Because the 3-letter is just the 3-letter words at the beginning of the amino acid. If I have asked you to remember the one-letter word, which I also do not like to remember, but it's kind of difficult to remember, then I will not ask you to remember the three-letter word. You get it? Everything is just about the amino acid, Jude. If you try to analyze the question, it may sound difficult, but if you have a good knowledge about the amino acid, then it is not a difficult question. So I hope you will do better in the next exam because I have more number in the next exam. Okay, so go ahead, Gwen. I'm done now. Gwen? Dr. Pipito, are you there? Nawala na siya.
Yes, Doc. Sorry, I was charging my laptop. Wait. Okay, so number 15 or the 15th question. This membranous organelle appears under the electron microscope with an outer membrane and inner membrane arranged in numerous crestae. Answer to this?
Mitochondria.
Okay, mitochondria. That's the correct answer. What is the function of this membranous organelle that appears as a stack of flattened membrane sheets, often adjacent to one side of the nucleus?
Golgi.
Golgi.
Okay, so the function is, the answer to this is modifying, sorting, and packaging proteins and lipids. Okay, this one, less than 30% got this one. So cell injury causes morphologic changes in the plasma membrane of the cell. What causes the formation of plasma membrane blebs? What's your answer?
Binding of microtubules, Doc.
So the answer to this is detachment of plasma membrane from actin filaments. Any question to that correct answer? Okay, I'll proceed. Chloroquine is a drug to treat and prevent malaria. This drug acts on the lysozymes in activating its enzymes. Membrane proteins in lysosomes are highly glycosylated on the luminal surface. What is the importance of these sugar molecules covering the luminal surface of the proteins? What's your answer? Answer is make the lysosomal membrane resistant to digestion by hydrolytic enzymes. Okay. Next item, smooth endoplasmic reticulum is a membranous organelle that is involved in detoxification of noxious substances. Which of the following best describes ribosomes associated with SER? Answer to this, SER has short anastomosing tubules with no associated ribosomes. So next item, mitochondria are abundant in cells that expend large amounts of energy. Which of the following statements best describes the structural component of the mitochondria? Answer to this?
Two membrane, Doc.
Two membranes, Doc.
So the correct answer is they possess two membranes. 21. These are membranous organelles that are single membrane bounded. In the liver, they are responsible for detoxification of ingested alcohol. What's your answer?
Peroxisomes, Doc.
Peroxisomes.
That's correct, peroxisomes. This is a product of cellular metabolic activity that is found within the cytoplasm. It is visible in the light microscope as a deep brown granule. It is mostly demonstrated in the spleen where aged erythrocytes are phagocytosed. What's your answer?
Hemosiderin.
Okay, answer hemosiderin. This one, less than 30% got this one. Mitochondria display a variety of shapes. Its structural components possess specific characteristics that are related to their functions. The enzymes of the citric acid cycle and fatty acid beta oxidation can be found in which specific area of the mitochondria?
Matrix.
Okay, so the answer is mitochondrial matrix. Which membranous organelle regulates programmed cell death?
Mitochondria.
Answer, mitochondria. Which of the following statements is correct on the difference of euchromatin and heterochromatin? So the answer to this one, as seen in the electron microscope, they both have granular filamentous appearance. However, heterochromatin is more tightly packed compared to euchromatin. Any questions so far? I'll proceed. In this phase of mitosis, initial separation of the sister chromatids happens. The chromatids are then pulled to the opposite poles of the cell. What's your answer?
Anaphase, Doc.
Answer?
Anaphase, no?
Anaphase. Okay. 27. How can you differentiate apoptosis from necrosis? Answer?
Necrosis is cell swelling, Doc.
Swelling, Doc, sa necrosis.
Okay, so the answer to this is cell swelling is evident in necrosis while blebbing is seen in apoptosis. Which of the following statements is correct regarding meiosis II? Answer, it produces daughter cells that have only haploid DNA content, 1D. Any questions for histology? None? So, yes, sir.
May I ask the other choices?
For this one, other choices, at its completion, the cell is diploid in DNA content, 2D. The chromosome number is reduced from diploid 2N to haploid 1N, and S phase precedes meiosis II. What's your answer, Cesar?
The question about with blebbing.
Sorry, I can't. You're quite choppy. What's your answer? Sorry.
The question about apoptosis.
Question on
Apoptosis, Doc.
apoptosis. Sorry. How can you differentiate apoptosis from necrosis? This question, yes, sir.
The other one, Doc, the first one.
What was the answer to that, if I may ask?
I think the detachment from actin filament, Doc.
That one. The membrane. Wait. Wait.
Number 17, I think.
Cell injury causes morphologic changes in the plasma membrane of the cell. What causes the formation of plasma membrane blebs? And the answer, other choices are polymerization of the cell's intermediate filaments are prevented, phagocytosis of the cell's plasma membrane's actin filaments, binding of the microtubules that supports the membrane. And the answer is detachment of plasma membrane from actin filaments.
Okay, Doc. So, Doc, there's no option about cell membrane alteration.
Sorry, Cesar.
There's no option about cell membrane alterations.
No option. No option to that one.
Okay, Doc. Okay, Doc. Thank you.
Okay. Any other before I proceed to physio and then the biochem part, the cellular activities? None. So I'll proceed. So you were given a figure, no? So note that water can only pass through the semi-permeable membrane. So if pressure is applied to the side with less solute, what will happen to the flow of osmosis? What's your answer to this?
Accelerate.
Accelerate, Doc.
So the correct answer is accelerate. With the same figure that you were given, know that water can only pass through the semi-permeable membrane if pressure is applied to the side with more solute. What is the likely flow of osmosis? What's your answer?
Stop. Stop, Doc. Stop.
The correct answer is stop.
Stop.
Okay. So next
Doc, may
question.
question ako, Doc.
Who's this?
Harvey Sarosa, Doc.
Sige, Harvey.
Doc, if I've applied the pressure on the right side with the more solid concentration, how does it stop, Doc? Does it not parang reverse or what?
So your answer to this is?
I answered stop, Doc, pero ano ang difference if the pressure would stop or reverse po, Doc?
I will let Dr. Sarita answer you that question to your emails. Okay? Or
Okay, thank you,
anyone
Doc.
with idea here? None? No one? Okay, sige. So 31. Recycling of organelles involves
Lysosomes.
lysosomes. That's the correct answer. Which best describes the sodium-potassium ATPase pump?
Near the sodium. Doc.
Okay, so the answer is ATPase located near the sodium binding site. So, what is an example of a symporter?
Sodium glucose.
The answer is sodium and amino acid transport. Which of the following is sufficient to make cilia move?
Axonym, Doc.
Axonym.
Answer, intact axonym. A 16-year-old female is diagnosed with Carter-Henry syndrome. Which of the following is a prominent feature of this syndrome?
Respiratory infection, Doc.
Recurrent pulmonary infection. So which best describes the relationship between flagella and cilia?
ATP dependent sila.
ATP, Doc.
So answer, both are ATP dependent for movement. Next, which best describes the sodium-potassium ATPase pump?
ATP.
Is it creates ATP, Doc?
So the answer is can create ATP with the appropriate condition. Okay, so you have another figure. So with that figure, which of the following will directly provide energy for the secondary active transport? Can you remember?
C. The one with C, Doc.
Cytoplasm.
C.
Cytoplasm.
C. So the answer to that is C. Which of the following, another figure, which of the following illustrates simple diffusion? What's your answer to this one?
A, Doc.
So, 11 more questions. What best describes the process of a macrophage engulfing a foreign substance?
Yung may maiwan na substances, Doc. I forgot the terms.
Glucose.
Yeah, that one.
Okay, the answer is phosphate and glucose diffuse freely from the lysosome to the cytoplasm. So last 10 questions. In phagocytosis, what process precedes the invagination and surrounding of the cell membrane around a targeted particle?
Chemotaxis, Doc.
Answer is receptors in the cell membrane attached to the ligand of the particle. Any clarification to this question? None. So clathrine is primarily used in the process of?
Pinocytosis. No?
So the answer is pinocytosis. Sige. So done with the official questions. Any clarifications? Concerns? Okay. Cell membrane. Which of the following is a mechanism for lipid asymmetry? What's your answer? So the answer to this is the enzymes involved in the phospholipid synthesis is located in the cytoplasmic side of the membrane. Why is this the answer? Lipid asymmetry, and then you have the enzymes in the inner usually, but then you have your flipases that can flip those phospholipids from the inner to the outer. So that can create your lipid asymmetry. Because the other choices, like the peripheral proteins, are usually located in the exterior, they pertain to peripheral proteins, not your asymmetry because of the lipids. Okay, they pertain more on, in general, your membrane asymmetry. But the question is specific for lipid asymmetry. Okay, now next question. Which of the phospholipids is prominent in myelin sheaths? What is your answer to this?
Sphingosine plus fatty acid.
So those are your
Fatty acid.
sphingomyelins, right? It's in your book, sphingomyelin, but your sphingomyelin is composed of sphingosine plus your phosphorylcholine, right? Your ceramide, sphingosine plus fatty acid, that's your ceramide, but it's not a phospholipid. Okay, letter C, fatty acid plus glycerol plus choline, that's your phospholipid. But the question is, a phospholipid prominent in your myelin sheaths. Okay? Sige. 45. Which characteristics of the membrane is due to glycoproteins? Very easy question. So your glycoprotein can contribute to, what's your answer to this? Asymmetry of the membrane, right? Especially your carbohydrate, glyco plus your protein. Okay, which is true of the characteristics of membranes? So very easy question. Answer. Transverse movement of phospholipids indicates its dynamic nature. Which of the following is true of integral proteins? So answer, they interact extensively with the phospholipids, especially the hydrophobic core. So which is the predominant molecule of a membrane? So very giveaway question also. Answer is phospholipids. Which of the following increases the fluidity of a membrane? Presence of more unsaturated fatty acids. Okay, increases the fluidity of a membrane. So this one question. At temperatures more than the TM, and then you have cholesterol, the fluidity of the membrane, what's your answer?
Decreases.
Decreases, Doc.
How about rigor? I will ask for rigor.
Maintains po, Doc.
Maintains. Okay, when you have temperatures more than TM, your fluidity increases, but you have your cholesterol as buffer so that your fluidity will maintain. So the correct answer to this one is maintains. Any questions? Sheralu has a so regarding the ATPase, if one of the choices is near the potassium binding site, wait, I'll go back to that question. So this is a question which best describes the sodium-potassium ATPase pump. So choices: 2 binding sites for sodium in the cytoplasm, 2 binding sites for potassium in the cytoplasm, ATPase located near the potassium binding site, and the correct answer is can create ATP with the appropriate condition. So
Excuse me, Doc. Can I ask lang, Doc, about the temperature, Doc?
Temperature.
Yes,
Last
cholesterol.
question.
I just have a question, Doc.
Yes.
So if if the temperature is above the TM, Doc, okay, what should be the fluidity day, Doc?
It increases, no, the fluidity. It can increase the fluidity of your membrane. It's more than the transition temperature. It increases the fluidity of the membrane. Increase in temperature, Queenie, can increase your fluidity, especially more than the TM.
Okay, Doc. Thank you, Doc.
Okay, sige. Sige, any other question? None? Okay, so you can, basing on your performance right now, you try to go through why you made those mistakes and improve on those mistakes for the next exams. So the coverage for this unit exam will be again assessed from you during the unit exam. But you have a unit exam long exam 2, Monday, that covers proteins, carbohydrates, and then lipids, right? So focus on those and then improve on your performance for the succeeding exam. Okay, any concerns? Then I can release you. You can take a break for a while, but tonight you have to read on your small group discussion for tomorrow. Okay? If none, no concerns, you can go.
Thank you, Doc.
Thank you, Doc.
Doc.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.